Hello, I'm Cindy Schoen, a rising sophomore studying math and statistics at Carnegie Mellon University and a 2021 summer trainee in the Biomedical Big Data Science program at the Mayan Lab. I will be presenting on the Augmented GSEA Enrichment Analysis Appyter. GSEA Enrichment Analysis is an analytical method for interpreting gene expression data. Before GSEA, expression analysis used to mostly focus on the differentially expressed genes between two phenotypes. But GSEA was created to go a step further and use these differentially expressed genes to detect underlying biological processes between the phenotypes through gene sets. It has since been used in various studies, including one about metastatic RCC, where it identified many chromosome maintenance genes as upregulated in RCC metastases. MCMs are important in cell proliferation and are prognostic biomarkers for poor outcomes in ovarian cancer. According to the paper, while they had been studied in other cancers at the time, they were novel for RCC research. Although there are variations, the standard GSEA method starts off by requiring a Wiener process walk down a ranked list of genes. The running sum is plotted, increasing if a gene in the list is also present in the gene set being analyzed, which is called a hit, and decreasing if it's not. The maximum deviation of this running sum from zero is the enrichment score. An enrichment score reflects how overrepresented a gene set is at the top or bottom of a ranked gene list. Next, the statistical significance or p-value of this enrichment score can be estimated using phenotype-based permutation testing, which preserves gene-to-gene correlations. Finally, there is adjusting for multiple hypothesis testing by normalizing the enrichment score and calculating the false discovery rate. These steps give us the four major statistics of GSEA that help us interpret the data. In this presentation, I will introduce the AGSEA Appyter, which performs and visualizes the standard GSEA as a web application, but also has an augmented feature that highlights novel genes that are potentially missing from the prior knowledge gene set and are also relevant to the input signature. For submitting expression data, the user can either choose to supply option 1, which is a pre-ranked list of genes, or option 2, an expression dataset with raw counts and a phenotype labels file along with a ranking method of the user's choice. There are 7 available ranking methods shown on the slide. The user must also submit a GeneSet library, which can be uploaded, or an Enricher library can be chosen. If an Enricher library is chosen, the button for applying augmented GSEA can also be toggled. The other available result options are choosing how many top gene sets the Appyter will display and how they are sorted. Tables and plots will only be generated for the top gene sets, but a CSV file with the full enrichment results will be downloadable. After going over the inputs, here's a case study so that we can look at the results and visualizations that the Appyter generates and interpret these results in context. This series contained expression data for independent biological triplicates of transformed lung alveolar cells overexpressing ACE2, which increases chances of SARS-CoV-2 infection, and they were either mock-treated or infected with SARS-CoV-2. The Enricher library chosen to use for AGSEA with this expression data was the KEGG 2021 Human Pathways. The chosen ranking method was characteristic direction, and the top gene sets were retrieved through sorting by p-value. The top 5 gene sets are displayed in a table for this case study. In the Appyter, this table is available for download as a PNG file, and the columns can be rearranged. A positive enrichment score implies that the gene set is upregulated by SARS-CoV-2, and a negative enrichment score implies that the gene set is downregulated. From this table, we can see that the coronavirus disease, ribosome, and steroid hormone biosynthesis pathways have positive enrichment scores, while the focal adhesion and leukocyte transendothelial migration pathways have negative enrichment scores. Now moving on to the enrichment plots for the case study. There is a drop-down menu not visible here for the user to be able to choose which gene sets plot they want to view, as well as multiple interactive features such as panning, zooming, and these hover labels. This figure shows all of the case study's top gene sets together. This slide has the individual plots for all of the case study's top gene sets and includes hit indices. Like the enrichment scores, the hit indices can also indicate whether the gene set is down or upregulated by SARS. In this case, if the hit indices are mostly on the left, the gene set is upregulated, and if they are on the right, the gene set is downregulated. From these plots in the previous table, we can categorize the top pathways into these 3 being upregulated by SARS and these 2 being downregulated. After performing standard GSEA, we can move on to explaining the augmentation feature and its methodology. By augmenting the KEGG pathway library, we want to identify missing genes that are relevant to the biological processes under investigation. First, augmented enricher libraries are produced by adding 100 top co-expressed genes that are retrieved using the ARCHES-4 gene co-expression matrix. This flowchart shows how that is done. Then, there are 3 random walks plotted, one for the original gene set, one for the augmented, and another for the 2 combined. The top co-expressed genes are augmented genes taken from the leading edge of the combined plot, and they are interesting because they co-express with the pathway and the input signature. This means that they are potential members of the pathways, but have not currently been identified. This slide shows the augmented plots for all of the top gene sets from the case study. Generally, because the augmented genes are co-expressed with the original, their hit indices are not too far apart. The augmented hits are shown in red, while the original hits are in blue. These are the tables for the case study's top co-expressed genes, and the tables all show up to the top 10 genes. Some of the gene sets have fewer than 10, which implies that there are not many genes missing from the original pathways. These co-expressed genes are predictions made by the Appyter for genes that play a role in the pathway but were currently missed in research. With these results, we can analyze if these genes have connections to the pathway whose genes that they were added to. The highlighted genes in red and blue will be talked about on the next slide. For this case study, we can take a look at these eukaryotic translation elongation factor genes, G and D, which are co-expressed with the ribosome and coronavirus disease pathways. Previous literature has shown that they encode subunits which are part of a complex that plays a role in replicating RNA viruses, like SARS-CoV-2. Since the ribosome pathway affects translation and replication as well, it makes sense that they would be co-expressed with both pathways. Meanwhile, the top co-expressed gene for the leukocyte transendothelial migration pathway that was downregulated by SARS-CoV-2 is MYH9, which encodes a conventional non-muscle myosin and has been shown by past studies to indeed play a role in neutrophil migration for immune response. The future directions of this project would be to decrease the runtime of the Appyter so that it works faster with bigger files, and adding an enrichment analysis report available for download would also be helpful for interpretation. Finally, augmenting more enricher libraries for more widespread usage and adding AGSEA to PRISM-X, which provides context-specific and better predictions, would also be important for the future. I would like to thank the Mayan Lab and everyone who has helped me throughout this project through the summer, especially Dr. Mayan, Sherry Shea, Minji, Daniel, as well as Sherry Jenkins for organizing our program. Thank you.